All right, peace be upon you all, and welcome to a new exciting session with Dr. Fuz Rana. Fuz, how are you doing today?
I'm doing great, Ahmed. How about you?
All is well, and now we are hitting the core stuff. What is the so-called central dogma of molecular biology? And the word dogma is actually very intriguing. So, is there a historical reason they just called it central dogma? Because dogma is typically not used in science, isn't it?
It isn't. And that term actually has its origin, interestingly enough, with Francis Crick, who won
Mhm.
the Nobel Prize along with Jim Watson and Maurice Wilkins for discovering the structure of DNA. And after he did that work, he spent quite a bit of time really playing a critical role in helping to define the principles of molecular biology. And
Yeah.
so he came up with this concept, the central dogma of molecular biology. And in an interview later on in life, he actually expressed regret about
Wow.
using that term.
Really?
Because yes,
I
because
didn't know that.
because in science, there is no such thing as dogma. There's theory and there's laws and things like that. And I think when he used the term, at least according to Crick, he argued that he was thinking that that term was approximately equivalent to a law, and then he realized the kind of the religious connotations later on. And Crick, was an atheist,
Yeah,
an
yeah.
outspoken atheist, by the way. But he also is well known for a famous quote that people who have a worldview like you and I, Ahmed, love to cite, and that is that biologists must constantly keep in mind that what they see was not designed but rather evolved. And this was an admonition that he wrote down in the very end of an autobiographical account of the discovery of DNA, What Mad Pursuit, which is a wonderful book. It's a charming story about, from his perspective, about the discovery of the structure of DNA. But it's interesting that even as an atheist, he was really acknowledging that the first reaction you have when you look at biochemical systems is that they look like they're designed, and
Yeah,
that
so but
as
but but
scientists
he,
we have to suppress that
but
that
he
that
said he said we must resist, yeah.
yes,
if
yeah,
if if it looks designed and feels designed and acts designed, it's still not designed.
yep,
That's unbelievable. So let's first see what he really found out, which is, I think, is still until today very good science, and
yeah,
investigations on the molecular level are confirming it. So
yeah,
let's go to your exciting
sure, sure.
presentation and understand what central dogma is.
Yeah, and we probably are going to be spending 4 episodes dealing with different aspects of the central dogma. And the reason why is because this is, I think, arguably the organizing principle in biochemistry. If there's an organizing principle, this very well could be it. And
Okay, okay. So
and
today we
and
are going to understand what it is, and then we're going to take it piece by piece, what
yes,
happens in the flow of information, etc.
yeah,
Okay.
and you're right. I mean, the central dogma is all about the flow of biological information. But the way I like to think about the central dogma is that it essentially is explaining how the cell produces proteins. And this is a cartoon that we showed in a previous episode,
Mm-hmm.
kind of giving us a sense of the structure of proteins. And proteins are these massive molecules that are formed from molecular chains that are built from amino acids that are linked together. And then these chains will fold, the backbone will fold into characteristic patterns, and those characteristic folds will interact to form the three-dimensional structure that can then interact with other proteins to form massive complexes that carry out virtually all of the operations inside the cell. Virtually every structure, every process in the cell has proteins that are contributing.
Mm-hmm.
And in fact, even the simplest cell has to have thousands of different types of proteins, many which occur as multiple copies of that same protein, in order for that organism to even be able to live. And so the question becomes, how does the cell know what type of protein to produce, and how much of that protein to produce? And the key to a protein structure is its amino acid sequence. And there are 20 different amino acids that are used to build proteins that are encoded within the genetic code. And it's the precise sequence of amino acids that ultimately determine the structure and consequently the function of proteins. So the question is, where is that information stored? And how is that information read and then used to make proteins? That really is what the central dogma is ultimately about. And
right.
And so, you know, we talk about the flow of information, and you know that information basically flows in this way, where the information to make protein is housed in the structure of DNA. And we talked a little bit about this when we talked about DNA data storage. And that information that's needed to make proteins is copied into the form of RNA molecules, where each RNA molecule corresponds to an individual protein. That RNA then migrates to a complex called a ribosome that reads that information in RNA and then is used to make proteins. That information is used to make proteins. And so this process is, again, the process of copying the information in DNA to make RNA is called transcription. And then the conversion of that information from RNA into proteins is called translation. And so right away, we begin to see the descriptors for these different processes, again, reflecting the fact that there's, again, information in biochemical systems, and that information is flowing in the way that we would make use of information, and the way we would handle information. You know, where the information in RNA has to be converted into a different language in terms of the information found in proteins, and that's why it's called translation. And so, you know, the analogy that I like to use, and there's different types of analogies that we could employ, would be like a library, where, you know, back in when I was in college, you know, if you wanted information, you had to really go to the library to get that information. And there were, you know, these bound volumes that would never be allowed to leave the library, right? Because they were such valuable sources of information that if you wanted that information and wanted to take it with you, you had to copy it either by writing it down on a piece of paper by hand or making a Xerox copy. That is, you were making a copy, you were transcribing that information. And then, you know, let's say that the volume that you were working with was in English, but you wanted to communicate that information into somebody that speaks Spanish. You would have to translate it then from English into Spanish. And that's exactly what's happening, you know, with this process, where, again, DNA's primary role is data storage, and it's the blueprint of the cell. You don't want that information, you know, handled improperly. You want to preserve its integrity. And so the way you preserve the integrity is it's only used as data storage. And when you want information in that DNA, you have to access it very, very specifically. You know, another example, and would be curious, you know, to maybe hear some of your analogies.
My analogy would be computer system based.
Okay, well, here's another analogy. Then I want to hear yours. But you know, I used to, prior to joining Reasons to Believe, I used to work in research and development for a Fortune 500 company. But even though I was in R&D, there were oftentimes that I had to interface with people at the manufacturing facilities. And there, you know, there are these making instructions that are held in the factory or the manufacturing facility's office. Those
Hmm.
making instructions are sacred. They never leave
The
the
recipes.
office. Right. Yes, because
Yeah.
if something happens to them, then we don't have the reference documents to tell us how to make the product. So if you wanted to take those instructions to the manufacturing floor, you would have to make copies of them, right? And
Uh-huh.
and then there would always have to be, the engineer would then have to translate those instructions into sets of, steps that machine operators or whatever might need to carry out. But
Yeah.
again, you're preserving that valuable information, and so the DNA is like the making instructions for the cell's manufacturing operations. You want to preserve its integrity.
Wow, that's also a very insightful thing from what we actually do as humans when there's a product to manufacture. Like, so there is the original design, and then there is a copy taken by the production engineer, and then he would give instructions to those on the shop floor. My analogy would be actually in computer systems. So if, for example, you have a blueprint for something that is going to be 3D printed by a 3D printer, let's assume that this is essentially the protein at the end of the day,
Mm,
then the design would be on a hard drive. Okay. And the reason it's on a hard drive is that you want it to exploit the properties of the hard drive, where, for example, bits will be stored in a magnetic medium. And because this is storage and you want to make sure that this storage lasts for some time, maybe you're using a storage cluster, for example, where you are at least having something like RAID storage, where each piece of information is stored on two different disks, so that if one gets corrupted, then you have at least another one to compare it to. And if they don't agree, then you know that the data has been corrupted. So even on our engineered systems, when you are storing something in terms of long-term storage, then you must have redundancy, which compares to me with the idea of the DNA being double-stranded. But then when you want to execute what's on the magnetic storage, you would, prior to sending it to the 3D printer, for example, you would read it into memory. So there will be correspondence between the bit that is on the magnetic storage and the bit that's on memory, but the medium is different. So
mm,
you will find, for example, that the medium, which is the computer memory, is quite volatile. If you lose power, everything in memory is lost, but everything on the hard drive is not lost. So what's in memory is typically very short-lived. You don't typically do multiple copies of it because memory is limited in size, but it has the property of being fast because it does not have to worry about all what the magnetic storage has to worry about. And then from what is in memory, you can now send it to the printer to be printed, and it takes shape. The 3D printer would print it in the way that it will become a physical product. Somebody can use it outside. And I find this to be very, very, very intriguing, that information in our cells are using those two steps. Because really the first question I would pose to you, Fuz, is why do we really need to have DNA and RNA? Why not just from DNA to proteins right away? So in our analogies, we understand why we do this. But is there really a reason that DNA versus RNA, there has to be this one extra step? So this is the question that I hope that this presentation will give us an answer for.
yes, and we'll get to that. We'll get to that in short order, because what you're getting at is that really the larger question, which is, does this central dogma have an undergirding rationale behind it, or is it just simply what evolution just happened to stumble upon? And that is actually a fairly common view, is that this process of the central dogma is the relics of the transition from the RNA world to the DNA protein world, where the idea would be that the very first biochemistry was RNA based, where RNA was functioning both as data storage, as well as the functional expression of that information. taking on the role of both DNA and proteins, and that later that RNA world evolved to give rise to the DNA protein world, and that the messenger RNA and even things like transfer RNA and ribosomal RNAs, which we'll talk about in future episodes, are just like the molecular fossils, the relics of this RNA world, regime. And so if that's the case, then there's really no rhyme or reason to what we're looking at here. But really the question becomes, is there a rationale? And we'll get to that in a second.
The why question.
The why question, right? Exactly. But now here's these are just some images that just help the viewer who may not be familiar with the Central Dogma, begin to have some again, better grasp of what we're talking about. So this is a diagram showing the production of the proteins at the ribosome, right? Where the ribosome is a manufacturing machine that is reading that information in RNA and is, again, producing proteins. This is a diagram showing the process in which that RNA is being copied, from the DNA double helix. And by the way, I think your analogy is probably a much more robust analogy than the two analogies that I, brought to the table. But, to me, I think what we don't want to overlook is not only is there, again, information that, speaks of the work of a mind, but the way that that information is manipulated, it's handled, the way that it flows, the logic that undergirds it is very similar to the type of way we would structure information, the way we would handle information, the logic we would use. And that to me, I think, again, points to the work of a mind.
Wow. Okay. So you agree that RNA looks like data in memory?
Yes,
and
I think that
looks like data on magnetic storage.
Yes.
You feel that corresponds to what is actually happening?
Yes, because if the viewers recall the episode we did on DNA data storage, remember we said there's all these features of the DNA molecule that ensure its structural integrity, its stability, that if there is any kind of mistakes that are introduced into that information, that there are ways that the cell's machinery can quickly detect those mistakes and correct them, right? Whereas RNA is really meant to be short-lived. It's copied, it goes to the ribosome, it's read, it's going to be quickly destroyed because you don't want the RNA persisting, because if it persists too long, you may end up producing more proteins than you actually need, or produce proteins that are, no longer needed at all, given the particular stage of the cell cycle that the cell is in. And so and there are actually structural features in RNA that distinguish it from DNA that actually make it more labile, that make it prone to degradation, that make it prone to being short-lived, right? And we'll see when we get to the episode on transcription that when DNA is replicated, there are all these systems in place to ensure the replication happens with a high degree of fidelity. When, and so this would be like making a copy of those manufacturing instructions, or the copy of that hard drive that you're then going to take to another location, to another facility. You want those instructions, you want what's on the hard drive to be error-free in terms of, that copying process, that replication process. But when you're with the RNA, the process of producing RNA doesn't operate with high fidelity. What's the point? Why put forth that effort? Because it's going to be short-lived. And if that particular copy is made with a mistake, you just make another copy. It's easy enough to overcome that mistake.
Yeah, so you make sure that you copy the blueprint with high precision. But if on the production line, this one new plastic 3D printed model is defective, then quality control can just throw it away or something.
Yes.
And we don't need to really worry about it.
Yes. Well, so it's interesting to me that, not to belabor this point, but it's interesting to me that that the analogies that just naturally lend themselves are, as you're saying, information system analogies or manufacturing analogies. And when we get into the process of protein production at the ribosome, we're going to see just these manufacturing analogies all over the place. Their manufacturing concepts become, very evident in the system.
Yeah.
Here's another slide that just shows how different regions of the DNA molecule, which are called genes, contain, the information needed to make the different types of proteins. Again, and so this is really what the central dogma is ultimately all about. Here's a diagram, another diagram showing the central dogma, where you have essentially DNA replication. There's the transcription process, and then there's the process of translation, making the proteins. Now, there's a wrinkle in the central dogma that it doesn't violate the central dogma, it just really expands it. Because you made a comment early on that really the ideas of Francis Crick are still valid today. And I think he produced the central dogma in the late 1950s, early 1960s. I don't precisely remember. And so this is an idea that's persisted for 60, nearly 70 years. So it's a robust idea, but there is a wrinkle to it. One is the discovery of an enzyme called reverse transcriptase that can convert information in RNA back into DNA that can then be reintegrated back into the DNA molecule. There are some viruses that have reverse transcriptase that plays a central role in their biology and in their replication cycle. And then there's also the discovery of what's called an RNA-dependent RNA polymerase, which can take RNA and make a copy of and make another RNA molecule, make a copy of itself. But it's actually a two-stage process because of the nature of how that replication works. So, initially the central dogma was that the flow of information is from DNA to RNA and proteins. That directionality is inviolable. And now what we see is that, well, you can actually go back from RNA to DNA or from RNA to RNA. And so it's really expanding the central dogma. It doesn't really violate it. And it's kind of a complex discussion as to why that's the case, but it has to do with the genetic code and the redundancy we see in the genetic code. And we'll talk about that in a future episode. But because of that redundancy, when you have a protein, you can never truly reconstruct what the actual gene sequence was. But if you go from DNA to RNA or RNA back to DNA, once you have RNA, you can reconstruct what the DNA sequence was. And so you're not really violating the central dogma or the directionality of the flow. What really establishes the directionality is the redundancy we see in the genetic code, which we'll again get to in a future episode. But anyway, this is just an expansion of the central dogma. That's the point of that slide.
Actually, I have a little bit of insight here from computer systems too.
Okay, yeah. Let's hear it.
Yeah. So while you're speaking, I'm thinking like this, because as far as I understand, reverse transcriptase is primarily used by viruses, as you just mentioned, and it just reminds me of computer viruses. So how do you typically get a computer virus? You typically get it maybe from the internet, maybe so it jumps from the internet into your computer memory. It was in memory somewhere, so it was in an executable format, because if it was sitting on a cold tape or something, it cannot go anywhere. So it needs to start in memory, okay? And then it will move from the memory of one computer that you are connecting to over the internet, for example, to the memory of your computer. And then what does it try to do once it's in your memory? If it just stays in memory, it goes nowhere, because once you close your computer, it's destroyed. Mm-hmm. So it immediately starts to try to store itself on your permanent storage. It looks for a USB drive. It looks for a magnetic storage. Maybe in the age of floppy disks, it will try to write itself on a floppy drive. It will try to write itself on your hard drive or on a USB. You know, that's why some companies would have a no USB policy. You cannot just plug your USB on the company's network, because then if there is a virus that has written itself to somewhere where it will then use it as a launch pad for further replication, it needs permanent storage. So the whole thing is, even before understanding that, when we wanted to have those parasites, okay, that will spread themselves, they need to start in memory so they can reach to somewhere. But while they are in memory, they are under the threat of being easily destroyed. They have to go back to permanent storage, just like, and exactly use this term, they try to hijack. They try to hijack mechanisms. They exploit some vulnerabilities in the system to write themselves back to the permanent storage, attach themselves to something on the directory structure of the storage, to a video, to something that is executable, to another exe or something, and then they use it from there. So even while you're talking, the very idea of reverse transcription, etc., and even the idea of RDRP, the RNA-dependent RNA
polymerase,
is just also what viruses try when they are in memory. They try to create many copies of themselves in memory, so to have more chances. If you find them and delete them, they can still have another copy that will eventually write itself to the hard drive. So amazing analogy to what an engineered system would be. Whether this engineered system is the good programs or the viral programs, unbelievable to even start to think that this is anything but the work of a mind. And just, I think even the research that is pretty recent, that viruses are not all evil. This is what I understand. Some of the viruses are just benign and they're just there doing their role and they're good. So
well, you know, I actually have maybe that could be a fun episode to do sometime, is why would a creator make viruses? Because I have a talk that I give from time to time, on that topic, because many people see the viruses as really undermining theism, because why would a all-powerful, all-knowing, all-good God create a world where there's this so-called natural evil, and there's actually very good reasons why viruses exist, and they play a very important role.
I think it is the case that we are getting in the way of viruses, not the other way around.
I would agree with that.
They just want to do their job.
Yeah.
And it reminds me, actually, when my kids would ask me, Dad, why are there flies, those things that bug us all the time? And this was what humans asked in the infancy of humanity. But later we understood that no flies is like, you'll not have any fruits, because those flies are everywhere pollinating and doing their job. Just keep your home clean and the flies will not come at you. That's it. We're just getting in their way. So it will be great if we really have something about those viruses. So we can put that in our second tranche. So let's go ahead. But it's really fun finding those analogies and how the mind of the creator is magnificent. It's only our miniature understanding that is making us have doubts. I think.
Okay, so now we've made some mention of the genetic code, but really, what we're talking about, with the Central Dogma is that the information which is in one format or one language, DNA and RNA, needs to be converted into another format or another language through the process of translation. And we talked in previous episodes that the information in DNA is essentially consisting of 4 subunit molecules, sometimes called the genetic letters A, G, C, and T, and it's the sequence of those genetic letters that harbors the information. Well, the information in proteins consists of 20 amino acids, and so how do you go from an alphabet of 4 to an alphabet of 20? And this is where the genetic code becomes important. And it turns out that what is used are called coding triplets or codons, which are combinations of 3 nucleotides, which gives you 4 to the 3rd power, or 64 coding units. That gives you ample capacity now to specify the 20 amino acids in proteins. And it turns out that because you have excess number of codons relative to amino acids, it allows you to specify the same amino acid with different codons or different coding triplets. And this is called degeneracy or the redundancy of the genetic code. And this is a very important feature that we'll spend a whole episode unpacking, because it's absolutely, to me, one of the most compelling reasons to think there's a mind behind everything is the nature of that redundancy. But this, again, is very much part of the central dogma, is that redundancy. And here's the genetic code, which shows the coding triplets and the different amino acids that those coding triplets, correspond to. But so not only do we have, again, the idea that there are analogies between how we, structure and manipulate information and handle information, between biochemical systems and human systems. But we also recognize that codes come from a mind. And when you talk about codes, there has to be agreement between the sender and the recipient, because the code is essentially defining the information. That information in DNA is fundamentally being defined by the genetic code itself. So this is another example of an analogy that we see, in the central dogma that again points to the work of the mind.
Yeah, and actually, I have an analogy exactly to computer code. Things like the ASCII code, or EBCDIC in the old days, where you want, actually, for example, you want to write the alphabet, okay, and then you have the ASCII code, which would assign to each letter or number or symbol an 8-digit code, which would be essentially, the codes would be only ones or zeros. Here the codes can be one of 4 things, and then the mapping would be 01001 is A, and then this is B, this is C. But here so you have the 8-bit will essentially give you the 256 possible characters that you can write. So also an amazing analogy. You have a limited number of possibilities for each bit, which is a 0 or a 1, but you put 8 of them together, and then they can correspond to a bigger alphabet of, in this case, just 3 places, and they correspond to 20. But then the redundancy here is amazing, and I'm excited to know your insights about that.
Yeah, and so just as a teaser, with something like the ASCII code, the assignment of the sequence of ones and zeros to A or B or C is essentially arbitrary, as I understand it.
It is arbitrary, absolutely, yeah.
Now, when it comes to the coding assignments in that we see in the genetic code, it's not arbitrary. There's some physical laws, of physics and chemistry, that are dictating the structure of the code, which is, and this to me is really mind blowing. But we're gonna, that's a teaser, and we'll set it aside for a future episode. But
that's amazing, because you're telling me that the why question can be even answered for that. But if you ask whoever have written the ASCII code, why did you assign the letter A that number, he would say, I just liked it or something.
Yes. Yeah.
Okay.
So anyway, a little teaser, so we can. All right.
Amazing.
So So now we're going to get to the question that really we all want to know, and that is, why the central dogma? Is there any rhyme or reason, to the central dogma, or is it just simply the artifact of some kind of, historically contingent evolutionary process, just the leftovers of the transition from the RNA world to the DNA protein world? Well, a number of years ago, I read a paper by a scientist named I.S. Dunn, and he wasn't really addressing the issue of the central dogma. But after I read the paper, I realized that the insight that he generated in this paper actually explains the central dogma. So let's take a look at some of the key points in this paper by I.S. Dunn, and then we'll look at what it means for the central dogma. And this researcher was interested in the question, what are the universal properties of living systems? Right? When we talk about life, is life on Earth just simply one of many examples of living systems, or is there something universal about life on Earth? And so, we've been talking about molecular alphabets.
Mm-hmm.
That the nucleotide sequence in DNA and RNA is a molecular alphabet. The amino acid sequences in proteins is a different molecular alphabet. But these alphabets are related to each other through the genetic code. And so Dunn was asking the question, because molecular alphabets play such a central role in biochemistry because of the central dogma, is this a universal property of living systems? And so he argued that you could never get the complexity you need for life just simply through the chemistry of small molecules, through small molecules that organize into autocatalytic cycles that might be integrated into very complex networks of autocatalytic cycles. As complex as those systems are, they don't really have the complexity you would need to support the operations of living systems. And then he also argued that if you had macromolecules, just a random collection of macromolecules, polymeric systems, that too doesn't give you the complexity that you need. What you need are actually macromolecules that are built from molecular alphabets, because that gives you, through combinatorial possibilities, the complexity that you need. So a small protein might have 100 amino acids in it. Well, with those 100 amino acid sequence, there are 20 to the 100th power possible combinations. So you through molecular alphabets, you get combinatorial possibilities that give you inherently the complexity that you need to support living systems. So his argument was molecular alphabets are probably a universal property of living systems. Now, the next point that he makes is that, molecular complexity can't be achieved with small molecules. You need macromolecules. Now,
but is that or not essentially contradictory to the theories about how life could have evolved from non-life? Because the arguments I understand in whoever is arguing for RNA world or RNA and peptides or whatever, they always refer to those autocatalytic cycles that then goes to something else. But
right,
what I hear you saying, according to Don, is that if it started this way, it wouldn't go anywhere.
That is an implication. You're exactly right, Ahmed. In fact, I didn't even think about that until you brought that point up. So I've been thinking about this paper for almost 10 years, and so, yeah, you're exactly right, though. This has profound implications, not only for what is the universal properties of life, but for different origin of life models. And so metabolism first models, as they sometimes are called, really are undermined by that particular insight. Now, this is where things get really interesting, because at least at this point, it still leaves open the possibility of the RNA world, but that possibility that door is going to be shut here in a second. Because
Wow.
what he realized is that when you think about, again, molecules made up of molecular alphabets, one of the requirements for life is that it's able to reproduce, which means that you
Mm-hmm.
need to make a copy. This is what DNA replication is. You need to make a copy of the molecules that are harboring the molecular alphabet. The problem is the process of replication puts constraints and demands on the system that mean that you actually undermine the what you gain by combinatorial possibilities. In other words, the constraints of replication actually uh, uh, take away some of that available complexity.
Uh-huh.
Because to replicate a system, again, there are constraints that you can't violate. Otherwise, that system can't replicate. So what he said is that you actually need to have a primary alphabet and a secondary alphabet, where that secondary alphabet is specified by the primary alphabet, but is not constrained by replication. Right?
Mm-hmm.
This is and so you need not only to have molecular alphabets, but there has to be primary and secondary alphabets. Well, this sounds very similar, does it not, to
Yeah.
the central dogma, right?
Yeah.
Where DNA is the primary alphabet, proteins are the secondary alphabet, right?
Uh-huh.
And how do you go from the primary alphabet to the secondary alphabet? You've got to have a decoding system, which would be the role that RNA is playing in terms of messenger RNA and then as well as transfer RNA. And you have to have a genetic code, a code that is defining how that transition from the primary to the secondary alphabet goes. So right away, this undermines the RNA world hypothesis, right? Because it's
Wow.
basically saying you've got to have a DNA protein world. But it also saying is that there is this elegant logic to the central dogma. It's not arbitrary, right? It's not haphazard. That there's no way to have primary and secondary alphabets unless you've got something exactly like the central dogma that is giving the overarching framework to how that information flows.
Wow.
And so this
So
is
it is like our DNA is built for replication and transcription, but RNA is built for execution. This
yes,
is what he's arguing. So you cannot have RNA as basis for storage because it's not made for
right,
that.
right. I mean, now in his paper, he doesn't talk about that the need for RNA as an intermediary, but once you start talking about primary and secondary alphabets, by definition, you've got to have that system, that translation system, which is
Yeah.
really what you're looking at where with RNA is
Yeah.
it's playing that intermediary role. So it's interesting to me when we talk about analogies between the central dogma and the reference books in the library, the manufacturing instructions in a plant, how you would make a three-dimensional object, with data stored on a hard drive. The reason why those systems are in place is because there's a logic to them.
Yeah.
And
Absolutely.
likewise, there's a logic to the central dogma. So the analogies that we see between those three systems and the central dogma are reflecting an elegant logic in how we handle information and an
Absolutely.
elegant logic in how the cell handles information. So
Absolutely.
it's not just simply an analogy that helps to illustrate the central dogma. It's an analogy that is deeper than that flows out of a fundamental logic to
Yeah.
how
Absolutely.
information needs to be stored and flowed. So anyway, I think this is absolutely amazing, because
It is.
it highlights the design that we see. It's not just the complexity of the central dogma, but there's a rationale, a logic, an ingenuity that I think really speaks to the work of a mind.
Yeah, yeah, sure, absolutely. And another example that popped into my mind, even if you're not looking at hard drives, even if you're looking at something like firmware in a machine, even very simple ones like calculators, etc. you would have something else, a different electronic component that stores the ones and zeros in a different format, something like, I don't know, an FPGA or a BIOS stored on a CMOS kind of chip, etc., which will have a structure that will enable it to faithfully preserve the firmware of the basic operating system of that machine. So you buy a laptop out of a store, and it at least has this program that is stored on a chip in a way that is only usable for storage. But once you boot it, it immediately starts putting it in memory, which has a different format, etc., and then it starts executing. So even in our systems that are old chips, you will have a chip for long-term storage, maybe has a backup battery or something to make sure that it doesn't fade away. And then the normal memory, like the RNA, and then whatever output goes to a 3D printer, goes to a screen, to a monitor, to a speakers or whatever, is there is always, again, the second layer of decoding from the memory to the final product, which for sure shows that this is absolutely engineered, as you see, because whenever we try to do something, we do actually the same, the same way.
Yeah. So So the thing that's amazing is that the more that we ask the why question,
yeah, there is always an answer.
The more that the more that we end up seeing. Sorry about this.
yeah, don't worry.
In my excitement, I yanked out my headphones.
Yeah.
But when we ask the why question, we just begin to see reason and purpose, and we're always rewarded by asking the why question.
Absolutely. And actually, another thing came to my mind now. I'm sure that you, more close to the biology, would have even an insight about it. It's like about the long-term and short-term memory of a human being. So I know we don't really understand how long-term memory and short-term memory works, but we know that in our synapses, long-term memory is stored in a different way. And then when we recall a memory, it goes to another place where it is current in our mind. And it also happens the other way when information is flowing in. It will go first to the short-term memory, and then we would store it, and it goes to deeper thoughts or not, according to other aspects, etc. But even this computer in our heads, if it's at all good to call it that, there is always a structure for long-term storage and immediate use kind of memory. And then out of this short-term memory, we can exhibit an output by speaking what we think or whatever, and we have this translation thing. So even the way our mind works is this way. There's always those three things: long-term kind of storage, short-term processing hub for it, and then an output phase or something. So I'm not sure if, as a biologist, you agree that this analogy can also work or not.
No, I don't know a ton about neuroscience, but what I do know, I would agree with your assessment, and I would agree with your analogy. I think it's I think it's a very good analogy.
All right. So is there something else that the Central Dogma will tell us? Do biochemists or molecular biologists, how would somebody escape from seeing a creator in that? It just perplexes me.
Yeah, well, again, I think it goes back to a point that you made in a previous episode that again I really think is profound, and that is most life scientists really don't ask the why question. They are consumed with the how and the what. And then when it comes to questions of origins, it's like, well, how can we explain how this system could have emerged through evolutionary processes? But by not asking the why question, you are operating within a framework of materialism, or methodological naturalism, which, is still a version of atheism in my view. where you're excluding any possibility that a creator has played a role. And so when you look at the central dogma superficially, the argument is, well, this is, an outworking of the RNA world. In fact, I've heard a number of original life researchers who have readily acknowledged that the RNA world hypothesis is just littered with all kinds of very serious, intractable problems, but they'll say it has to be the RNA world. And because the only way you can escape what's called the chicken and egg paradox is through the RNA world. And they would say, and look, we have these molecular fossils in biochemistry, where RNA is serving this intermediary role between DNA and proteins. And so they, by not asking the why question, they just simply see this as a system that is inelegant, that it's just something that's been cobbled together, and that the complexity isn't an elegant complexity, but it's just something that evolution, again, produced. But when you ask the why question, and honestly, even I.S. Dunn, who we discussed, the scientist whose work we discussed, he wasn't even asking that why question. He was just trying to assess whether molecular alphabets are part of a universal feature of life. It was, when I read that paper, it was like, you have just described the central dogma, and have explained to us why there has to be a central dogma.
Absolutely. Actually, I have this argument against the RNA world I would like to share with you. Maybe you correct me if I'm wrong. In one of those episodes I have on this is evolution true or false course, I go like this. Let's assume, I'm putting a hypothesis. Let's assume that RNA world is correct, because evolutionary chemists and evolutionary biologists are obsessed with it. So I say, let's assume it's correct. What would be a prediction? So I go like this. If life persisted for long enough as RNA-based life, so genetic material is on RNA, we should expect today to find living systems that are RNA-based and other systems that are DNA-based. Because when you're talking to an evolutionary biologist and you tell him, if human evolution, why apes? He would laugh at you, okay? Because he tells you, if immigrants to North America are initially British, it doesn't mean that there will not be any more British, right?
Yeah.
So, okay, let's even accept that. So in your mind as evolutionary biologists, you would say, if RNA, you have the same logic. If RNA world is true, then we should, and then it moved to DNA, then it should not wipe out the RNA-based organisms. Then we should have at least unicellular life, which is the most versatile kind of life on Earth. Still, we can find living cells, not viruses, real living cells that are RNA-based, but they're nowhere. I did all the research that I can do until I found an exclusive statement that every single kind of life on Earth today, a living cell, has to be DNA-based. And the central dogma is applicable to every single living system. Yeah. So then I go, then if the hypothesis gives a prediction that proves false, then the hypothesis must be false. What do you think? Would it be a fair prediction to say that if RNA world hypothesis was true, then we must find RNA-based life? And if this is the case that there isn't this also a case for sort of irreducible complexity, that the system has to occur in the way that Don has predicted, which then says that somebody must have put it forward as a complete system at a point of time? Is this a valid argument in your opinion?
Yes, it is. I think that's a really important insight from your perspective, because you're right. You would expect to see, particularly when you think about the extent of the biodiversity in the microbial arena, with archaea and bacteria, that you should see, some organisms that would be exclusively RNA based. And you should also expect to see kind of the transitional forms where you would have organisms that would rely to a greater or lesser degree, on the central dogma. But to your point about the fact that this is kind of an irreducibly complex system, I would agree with it. That when you think about the central dogma, it is irreducibly complex. It all has to be in place for the central dogma to work. You can't have a partial genetic code. You can't have partial systems for transcription or translation. It all has to be not only there, but has to be, in its final form. And this is really part of the challenge of the chicken and egg paradox for original life researchers is, DNA and proteins are mutually interdependent. And so you can't escape that, right?
Yeah.
And the only way to escape it is the RNA world hatch, and that just it doesn't work for a number of reasons, from so so it doesn't work for a number of reasons.
Yeah. So okay. So I hope that one day they will give up on the this false. I'm glad that they're still calling it a hypothesis. I have run into people who call it the RNA world theory. I say, guys, this is just a hypothesis, please. And it's obviously false. But all right. So, Fuz, thank you very much for this great explanation for the central dogma. I'm sure this is a great foundation to going through it piece by piece on the coming, I would say, 3 episodes. So we are going to go replication and then transcription and then translation, right?
Yes, that's the plan.
Absolutely. So everybody, be sure to see this one and share it with your loved ones in preparation for the coming 3. And if you have had some questions on the live chat, make sure to join us on the after show that you will find a pin to it in the live chat, so you can go there and see you on the other side. Fuz, this has been as exciting as every time. And thank you very much for this. Hello? Fuz?
Yes. Yes.
Yeah, I'm saying thank you very much for this. It has been as exciting as every time. And I'm so excited to hear you answer the questions on the after show and to the upcoming ones too.
Yes, I can't wait.
Thank you very much. And to everybody, peace be upon you all. And see you on the other side in the after show and also on the next episodes. And please share this series with your loved ones. And peace be upon you all.